enjoying the second day of MDF's first virtual conference. I'm Dr. Nadine Ann Skinner, the Research Coordinator at MDF, and I'm excited to share that it is time for our industry partners to talk about their myotonic dystrophy programs and their breakthrough work in the development of different treatments. This is a chance for us to hear directly from these companies about their approach, their process, and their progress. Today we'll hear from Syros Pharmaceuticals, AMO Pharma, and Harmony Biosciences. We will not have time to ask the speakers questions during this session, but many of our industry partners have exhibit booths, which will be open for live chats now through 6 p.m. if you have follow-up questions. After the session, just go to the Exhibitors tab on the menu bar of the conference website, select Conference Exhibitors, and click on the company you're interested in connecting with. Once you're on the company's page, there'll be a button that says Join Live Showcase Now. Click on that button to join the conversation with that company. And when you're done with that, don't forget to join the dance party this evening too, for disco or rumba, or to dance to your favorite songs. Thank you for attending the conference and enjoy the presentation.
And welcome to an update from AMO Pharma. My name is Ibraheem Mahmood and I'm the CEO of AMO Pharma. But before I get stuck in, I'd like to take the opportunity to thank MDF for putting on this event and for giving me the opportunity to talk, and for you, the audience, for giving me some time with you today. AMO Pharma is a small team. We're just 8 folks based in the UK and the USA. But I must say I'm very, very privileged to be working with this team of hugely experienced people at developing new medicines for the treatment of child-onset neurological disorders. The team is applying its experience at working with groups like FDA to develop a program we're running focused on congenital myotonic dystrophy. Our team isn't just professional. Many of us also have a personal interest as we have a family member with a developmental disorder like myotonic dystrophy. This matters to us. AMO Pharma is developing a new experimental medicine called AMO-02, sometimes called tidaglutide. AMO-02 is an investigational medicine that is currently not approved for prescription for any medical condition. The medicine is prepared as a strawberry-flavoured liquid that is taken once a day by mouth. We are interested in AMO-02 for several reasons. First, because the medicine has already been tested in a variety of conditions in many hundreds of patients for up to a year, we have clear evidence that it is generally a safe and well-tolerated medicine, which is a great place to start when investigating a new medicine. Secondly, our study of AMO-02 has shown that it reduces the type of RNA in muscles and in the brain that leads to myotonic dystrophy. Here are some pictures that show the effect of AMO-02 on the RNA that causes myotonic dystrophy. The red on these diagrams is a special stain that highlights the presence of this RNA. The 2 pictures at the bottom are in a mouse that was engineered to have a lot of this particular RNA, and the 2 pictures at the top are from the muscle of a child with myotonic dystrophy. In both cases, we can see the presence of this RNA in red in the pictures on the left. But on the right, when AMO-02 is added, the RNA disappears, both in the mouse and in the child with myotonic dystrophy, which makes us hopeful that this drug will be useful for folks with myotonic dystrophy. We'd like to thank Professor Tinchenko and team for doing this groundbreaking work with us. So, in short, we have good evidence that AMO-02 is generally safe, well tolerated, and that it removes the RNA that causes myotonic dystrophy. So our next step was to test AMO-02 in patients with myotonic dystrophy. This is what we call a phase 2 study, the first clinical trial of an investigational medicine when it is given to patients affected by a particular disease. The aim of a phase 2 study is to see if an investigational medicine is safe and well tolerated in the patient group, but also to explore if it reduces the symptoms of their disease and how best to measure that improvement. In this case, we ran a phase 2 clinical trial in children and young adults with congenital and juvenile onset myotonic dystrophy, with the main aim of showing that AMO-02 was safe and well tolerated in these folks with myotonic dystrophy. We also wanted to rehearse how best to measure any changes in their myotonic dystrophy and using those measures see if they improved. In this phase 2, which was run by Professor Lockmuller and team, AMO2 was given once a day for 12 weeks while the disease was measured in various ways to see if it was making a difference in the patients in the clinical trial. For example, in this diagram, you can see a measure of the disease developed by Professors Heatwell and Johnson, which they adapted from advice from the FDA for use in clinical trials of myotonic dystrophy. Remember, we are looking at the effect of AMO2 given once a day for 12 weeks. We actually gave AMO2 at 2 different doses, a lower dose of 400 milligrams and a higher dose of 1,000 milligrams. You can see as the 12 weeks of treatment with AMO2 progressed, the symptoms of congenital and juvenile onset myotonic dystrophy improved. In fact, if you break down what was measured here, you would find that symptoms improved in a wide variety of symptoms, including in cognition, in myotonia, in mobility, in gut, and in pain. This meant that we saw patients soiling the bed less often at night, we saw patients with improved speech, and we saw patients more able to get around. In summary, when we consider all the data obtained from this study, our feeling is that AMO2 did indeed reduce the severity of symptoms of congenital and juvenile onset myotonic dystrophy. We're obviously hugely excited about this, and we would also like to thank Professors Lockmuller and team and all the families involved who worked so hard to complete this study. On the back of this result, we have decided to move to the next and hopefully final stage of testing and run a phase 3 study. Such a phase 3 study, if positive, should be the final step in developing a potential medicine for doctors to prescribe. Our plan is to run a phase 3 study of AMO2 in children and adolescents with congenital onset myotonic dystrophy. This study has been designed with advice from doctors who are experts in this condition and with advice from the FDA. When the study starts, it will involve 5 months of treatment and will be conducted at sites spread across the world. We had planned to start the study earlier in the year, but we were interrupted by the COVID-19 pandemic. Safety will always be our main concern, so that we thought it only appropriate to pause the study while we thought through how to conduct the study in a COVID environment. We have taken a lot of advice from doctors and worked with FDA extensively to design, with modifications, to make the study safer for patients, caregivers, and doctors. Our goal is to have this study underway from November this year in regions of the world where it is safe to start enrolling patients. As sites open for enrollment, we will announce them, and we have also launched a website dedicated to this study that will list sites that are ready to accept patients into the study. That concludes our update for now, but I very much hope that working together there will be more good news to come. Until then, stay safe and goodbye for now.
Hello, I'm John Carulli, Vice President of Biology at Syros Pharmaceuticals, and I'm very happy to take part in the Myotonic Dystrophy Foundation 2020 virtual conference. I have a short slide deck that I'd like to share with you to tell you about Syros Pharmaceuticals and our Myotonic Dystrophy drug discovery program. Syros Pharmaceuticals is a relatively young company. We were founded in 2013 with the goal of developing small molecule drugs to control gene expression for therapeutic benefit. We have a deep pipeline of gene control drugs, including SY1425 and SY5609, which are both in clinical trials for different kinds of cancer. We also have a drug discovery pipeline that includes other cancer projects, as well as genetic diseases, including sickle cell disease and myotonic dystrophy. Our myotonic dystrophy program right now is focused on DM1. and we're working on a small molecule for oral delivery. Small molecules can distribute to disease cells throughout the body. Our approach complements the antisense and gene editing approaches being taken by many other companies, and we hope to provide more options for DM1 patients. We're currently in drug discovery. That means we're identifying new drug targets, we're developing new drug screening tools, we're evaluating current Syros small molecules for their effect on DM1 models, and we're developing completely new chemicals to evaluate their effect on DM1 models. As you know, DM1 affects multiple organ systems in the body, including the neuromuscular system, the cardiovascular system, the gastrointestinal system, and others. Our gene control drug discovery program aims to inhibit or shut down production of toxic molecules from the disease-causing gene. DM1 is caused by a mutation at the DMPK locus on chromosome 19. Specifically, this mutation takes the form of an expansion of the sequence CTG in the DMPK gene. When this gene is transcribed into RNA, you get a CUG expansion in the RNA, and this CUG expansion assumes the secondary structure in the nucleus of the cell. And when it does that, it binds up and sequesters RNA processing proteins that are supposed to be doing other things, including proteins such as muscleblind. When this happens, that leads to downstream effects like the missplicing or misprocessing of different RNA molecules, as shown here in this figure from a paper published a few years ago. In 2017, Charles Thornton and Eric Wang and colleagues described multiple approaches to developing drugs for myotonic dystrophy in this review article. And these approaches include transcriptional silencing, or inhibiting the production of the toxic molecule from the DM1 locus, or post-transcriptional silencing, which might take the form of eliminating the toxic molecule once it's produced, or otherwise blocking that molecule. They described other methods as well. In principle, the Syros technology can apply anywhere in this process, but right now we're focused on transcriptional silencing. Our program fills fundamental gaps in the DM1 drug discovery landscape. With our focus on transcriptional silencing, we're capitalizing on our core expertise in regulatory genomics and gene control drug discovery. We're combining cutting edge with classical technologies to maximize our opportunity to discover disease-modifying therapies. We have partnerships with different academic groups, including Andy Berglund at Albany, to access their disease models and drug screening technologies. In Syros Labs, we're using genome engineering technologies to develop patient-derived cell lines suitable for high-throughput screening. We're also developing high-throughput imaging and other molecular technologies to support drug screening. I'd like to tell you a little bit about what we're doing in our labs. Starting with cells from DM1 patients, we've used genome engineering technologies such as CRISPR-Cas9 and others to develop cell lines that stably produce disease features and that do that stably across the cell cultures, different cells within a petri dish, and also over time as the cells divide. Some of those features include these ribonuclear foci that I discussed earlier, and those are those white spots within the large blue nuclei that you see on the right. And it also includes the missplicing that I described, as can be seen in the lower right of this figure. Once we have a few of those cell lines, we can benchmark them against reference molecules. To do this, we use tool molecules published by other teams. These are molecules that have a beneficial effect on myotonic dystrophy features in cell lines or in animal models, but that haven't reached clinical approval for a variety of reasons. These are still extremely useful molecules for characterizing cell lines and for understanding DM1 biology. What you can see in the lower left is that when we treat our cells with actinomycin D, previously shown by other groups to reduce production of the toxic molecule from the DM1 locus, we get the same result. As you go to increasing doses of actinomycin D, you get less production of the toxic RNA molecule, as indicated by the dark blue bars in the left-hand graph. In the right-hand graph, you see some results of treating those cells with an antisense oligonucleotide. This antisense oligonucleotide, again, was shown by others to reduce the amount of the toxic DMPK RNA, as shown here in the light blue bars in the right graph, and to restore proper splicing of some of the targets, as seen in the dark blue bars going up with increasing dose. What these results tell us is that our cell lines are really well suited for small molecule drug screening. They have the CTG repeat at its native locus in the genome, which is important because that means it's surrounded by all of the gene control elements that normally control expression and where we might be able to intervene with small molecule drugs. These cells also have the RNA foci that I described, as well as the splicing changes. Critically, these features are stable in cell culture. We've developed medium to high throughput assays to evaluate the effect of small molecules on those features, and we've shown that they're responsive to reference molecules. So these cell lines are ready to go for small or large scale screens of small molecule drug candidates. I won't have time to tell you any more about the details of the Syros Myotonic Dystrophy Drug Discovery Program, but I'll use this generic drug discovery funnel to let you know where we are and where we're going. This funnel starts at the top with target discovery and high throughput assays such as the ones I described earlier, and it ends at the bottom with clinical testing and hopefully clinical approval. Syros is at the top of this funnel. We're developing new screening tools, we're executing small molecule screens, and we're doing deeper evaluation of some molecules that showed desirable properties in our initial screens. While this project is at the top of the funnel, the Syros team has delivered molecules to the clinic for multiple programs, and we're well equipped to drive this program all the way to the clinic. Here's the Syros gene control pipeline again, with that figure from the Myotonic Dystrophy Drug Discovery publication. And the point I want to make here is that Syros is well suited for myotonic dystrophy, and myotonic dystrophy is well suited for Syros. In summary, the Syros DM1 program uses patient-derived data and cell lines to accurately model human disease biology. It capitalizes on Syros' expertise in gene control biology and chemistry. We're focused on orally available small molecules. We're leveraging existing Syros chemical assets to explore how they affect myotonic dystrophy cell models and eventually animal models. And we're also looking for new chemical matter to develop myotonic dystrophy small molecule drugs. Importantly, thanks to all of you, our work feeds into a well-established translational and clinical ecosystem that's ready to test these molecules in the clinic when the day comes. Thank you for listening. Please feel free to reach out to me anytime, and please learn more about Syros by visiting our website. Have a great day.
Hello, I'm Dr. Bill Jacobson, Senior Director, Clinical Development at Harmony Biosciences, and I'm a member of the team that is currently exploring a clinical development program to study non-neuromuscular symptoms in patients with myotonic dystrophy. Harmony Biosciences is a publicly traded pharmaceutical company that is headquartered in Plymouth Meeting, Pennsylvania. We work on novel treatment options for people living with rare neurological diseases, and our initial focus is on disorders of sleep and wakefulness. Patients are at the heart of everything we do, and we put breakthrough science within reach. We strive to advance the understanding of rare neurological diseases and to provide information and resources to people who live with and healthcare professionals who treat people living with such disabling, debilitating neurologic disorders. Today, I would like to talk to you about our efforts in myotonic dystrophy, or DM. As you're all aware, DM is a multisystemic disorder which impacts very many different body systems. Much attention is focused on the primary muscular symptoms of the disorder: myotonia, muscle weakness, muscle atrophy, smooth muscle involvement, and so on. The disorder, however, also does manifest itself in several other body systems, including cardiovascular, endocrine, and others. Harmony is focused on the central nervous system, or CNS, and its involvement in DM. There are several ways in which this is manifest, including excessive daytime sleepiness, fatigue, and some degree of cognitive impairment. Excessive daytime sleepiness, as well as fatigue, have both been shown to be among the most frequent complaints in patients with both DM1 and DM2, and both have extremely high impact on the lives of individuals with these disorders. Often, there is an involvement of sleep-disordered breathing, such as obstructive sleep apnea. Standard patient care treatment for obstructive sleep apnea includes positive airway pressure, or CPAP. But many studies have shown that while such treatment may address the breathing disorder, it does not necessarily improve the daytime sleepiness. Additionally, the degree of daytime sleepiness, which can be very disabling and disruptive to one's functioning, does not correlate with the degree of muscular involvement. Fatigue is also a problem in DM. Fatigue and sleepiness are related, but they're not the same thing. Sleepiness is the extreme desire to fall asleep or the inability to stay awake, even in situations in which wakefulness is required, such as at work or behind the wheel of a car. Fatigue is a state of overwhelming, sustained exhaustion and decreased capacity for both physical and mental work, and it is not relieved by rest. Both excessive daytime sleepiness and fatigue are common features of DM. Cognitive impairment is also a common feature of DM. Much of the work that is ongoing to develop therapies for DM focuses on the neuromuscular symptoms of the disorder. Harmony is interested in studying non-neuromuscular symptoms. Excessive daytime sleepiness, as well as fatigue and cognitive impairment, represent a substantial burden of disease, and the cognitive symptoms may increase as the disease progresses. There are a number of different cognitive domains involved in DM, and these include problems with such areas as verbal memory, visual attention, speed of processing, and executive function. People with DM often complain of a brain fog that makes it difficult to concentrate and function. The drug that we are investigating is pitolisant. Pitolisant was approved last year in the United States as Wakix for the treatment of excessive daytime sleepiness in adult patients with narcolepsy. Pitolisant works to increase the amount of histamine in the brain, a neurotransmitter that promotes wakefulness, and it works differently than a traditional stimulant. We are planning to investigate how pitolisant impacts excessive daytime sleepiness, but we will also be examining its impact on fatigue and cognition. We feel there is a strong scientific rationale to study pitolisant in patients with DM1 and DM2, but first must get the okay from the FDA to open up our clinical development program. At the present time, there are no approved therapies to treat these non-neuromuscular symptoms of DM. Stimulants such as amphetamines and amphetamine-like compounds such as Ritalin, caffeine, selegiline, and modafinil have all been used off-label with mixed results. There remains a large unmet medical need to address these aspects of the disease. Harmony is currently working with the FDA to open an investigational new drug application, or IND, after which we plan to begin a phase 2 clinical trial. We will share more information with the DM community when we are able to. I also hope to be able to join you in person in 2021 in San Diego and to provide you with an update on our clinical development program in DM. On behalf of Harmony Biosciences, my thanks to the Myotonic Dystrophy Foundation for the opportunity to speak with you today. And I want to thank all of you for your attention and wish that you all remain safe and well. Thank you.